Perfect. Good evening, ladies and gentlemen, and welcome to this webinar from DSM and Biomin about antimicrobial reduction. It is undisputed that any use of antimicrobial substances leads to resistant development in microorganisms. This is not only applied to antibiotics, but also to other antimicrobials, such as disinfectants. The underlying resistant microorganisms or their gene can nowadays be detected also far away from the application environment. If this topic is left overlooked, the consequences for humans and for the planet could be severe, especially if you're looking at an increasing population that has a higher demand for meat and proteins in general. When it comes to our industry, we are constantly receiving invitations to webinars where this topic is mentioned, together with solutions to mitigate it, especially during the pandemic. Every company is having a webinar on this topic. But when we talk to animal and feed producers, we can clearly see that a full understanding is not there yet. This is actually what gave us the idea to set up this webinar. We didn't want to go for the usual presentation, but we wanted to collect your burning questions on the topic and answer them informally in front of you. This means that today you will not hear a presentation or you will not see trial results. Today you will hear your questions answered in a simple and pragmatic way. My name is Michele Muccio, and I am your host for today. I have the pleasure to have in the call with me 3 experts in the field, covering different aspects of feed additives. The first speaker is Dr. Maya Segura Wang. Maya holds a PhD in molecular biology and has worked in different fields of research, combining experimental and computational approaches to study genetic variation. She joined Biomin 3 years ago as a bioinformatician, and she's been using high throughput sequencing approaches to understand the dynamics of complex microbial community, known as the microbiome. And also she worked on antibiotic resistance. The second speaker is Dr. Laurent Gabardo, and she is an expert in mycotoxin risk management. Laurent is a veterinary and holds a master's degree in veterinary science by the UFPR in Brazil. She worked for 4.5 years with feed additives in monogastric species across Latin America. In 2020, she joined Biomin DSM as a global product manager for Mycotoxins, and she's currently based in Austria. Her expertise in poultry gut health, feed additives, and strategic marketing as well. The third speaker is Dr. Neil Gannon, swine nutritionist and expert in phytogenics and probiotics. Dr. Neil Gannon was born in Sydney, Australia, and completed his Bachelor of Agricultural Science at the University of Sydney in 1986, majoring in animal production. He then studied for his PhD at the University of Sydney, and afterwards he completed a postdoctoral fellow at the Baylor College of Medicine in Houston, Texas, United States, where he investigated the impact of weaning on protein metabolism and gut function in piglets and humans. Dr. Gannon joined Biomin Australia in 2011 and as managing director with an active on-farm role in providing nutritional solutions to the larger pig, poultry, dairy, feedlot, and aqua businesses in Australia. In October 2015, Dr. Gannon commenced his role as a regional product manager for gut performance in Asia Pacific. This is the panel of today. I want to start immediately this talk. As I mentioned before, this is an informal talk, so we have no plans. We haven't prepared anything. There will be no presentation, so we will just go over the questions. But before we start, I would like to ask Dr. Gannon for his to make a small introduction on the topic of antimicrobial resistance. I believe, Neil, you have a few slides, a couple of slides that you want to show, and those are pretty much all the slides that you will see in today's webinar. So over to you, Neil. I will make you a presenter.
Thank you very much, Michele. Yes, and welcome everybody. It's not a formal presentation, but just to set the scene, I think it's important that we just quickly review what it is in terms of microbial resistance that we're talking about, because many of these concepts I want to just briefly introduce at the start will become relevant when the three of us start answering the questions that you have, as an audience, have posed, and we really appreciate you answering the questions. Just quickly, the mode of action of antimicrobial agents is quite varied, and when we look at the typical antibiotics that are used in our industry, you can see all the different names on this slide, and there's three main areas that these antibiotics target. They can either target the DNA, they can target the ribosomes, or they can target the cell wall. And depending on what those compounds do, they will disrupt the metabolism of that bacteria and ultimately kill it. And so when we start looking at the mode of action, we can start to see that depending on the antibiotic, there are many different mechanisms of antibiotic resistance. And even though there's a couple of mechanisms, there's actually thousands of variations. And this will become obvious in the coming dot points. So the bacteria could produce a destructive enzyme to neutralize the antibiotic. The bacteria could modify the antimicrobial target, so by mutation, so therefore the drugs cannot recognize that particular target, and therefore it becomes resistant to that antibiotic chemical. The bug can also prevent the antibiotics from entering by creating a biofilm or otherwise reducing the permeability of that cell wall. The bacteria can increase its pumping out activity, so it can remove the antimicrobial agent by pumping them out should they get inside the cell. Or the bacteria can create bypasses that allow the bacteria to function without the enzyme targeted by antibiotics. And when we look at the general class of antibiotics, they are largely a single chemical, maybe two combinations of antibiotics used, but they're very specific chemicals and they will target one of these particular activities. And so this is why antimicrobial resistance can develop quite quickly, because the bug only has to get around one particular chemical. Importance in terms of resistance, and when we look at what goes on inside a bacteria, we have the DNA, but more importantly, there's fragments of DNA, and again, this is a very simplistic overview, but fragments of the DNA encapsulated in this plasmid. And this plasmid can be excreted by this bacteria and picked up by another bacteria of the same family or any other bacteria from different families. And this is the concept that we really have to understand in terms of antimicrobial resistance. So this could be an E. coli, it could excrete that plasmid, it could be picked up from a salmonella or any other bacteria that's in the environment, and therefore all of the genetics coding for resistance, which Maya will talk about a lot more in this coming talk, will be conveyed to that new bacteria. And why does this become important? Well, when we start looking at antimicrobial resistance, this schematic here just gives you an indication of the pathways where antimicrobial resistance, these plasmids, can be transferred around the environment. And we are focusing on this top area here in terms of antibiotic use in animals and how that antimicrobial resistance can be transferred to the workers. But we also know that food, the crops, the animals that eat the crops, these plasmids can get into the water supply and become another source of food. But there's also, we can swim, we have wild animals defecating into that water source, we have our companion animals, and there's lots of different ways that these resistance can be transferred. We also know, as we've seen with the COVID crisis, that with the advent of fast travel through planes, resistances can be transferred rapidly around the globe very quickly. And at the heart of the situation about AMR is that if people get sick, get taken to a hospital, we have no antibiotics that will work against these bugs. And it is a serious issue in terms of preventing us as humans from dying. Similarly, if we have sick animals, we would like to be able to treat our animals with antimicrobials to get them healthy as well. And we want to preserve our antimicrobials to do that. And this is what's at heart of our antimicrobial reduction. We want to have the prudent use of antimicrobials, and we know that we can preserve their activity for as long as possible. Because at the end of the day, there's not large pharmaceutical companies investing in antibiotics because resistance becomes developed quite quickly. And so as Professor Mary Barton, a well-known academic in this area, said, we all swim in the same gene pool. And therefore, reducing antimicrobial resistance is every industry's problem, and it's an opportunity. It's not just the animal industry, it's not the companion industry, and it's not the human health industry. It's all part of us, and this is around the world, we have these One Health programs. And finally, I just wanted to talk about the number of plant secondary metabolites that are available or that have been identified. And you can see in terms of their simple chemical structure, whether they have nitrogen or without nitrogen. And there's literally thousands of products that have been identified in the plant secondary metabolites, all the herbs and spices that we've been consuming as humans for many years. We know that these compounds have great activity around antimicrobials. And with such a diversity of components and range in inclusion levels needed to have an effect, the probability of bacteria developing a resistance to a phytogenic feed additive is going to be very, very low, because we're dealing with many substances here, not just one or two defined chemicals as in an antibiotic. And with that, Michaela, I'd like to hand it back to you to continue on with the panel discussion. And hopefully that background just sets the scene a little bit about why AMR exists and what are some of the strategies that we will be using and discussing in today's talk. Thank you.
Thank you, Neil. That was a very, very simple and clear introduction. To be honest, I don't think it gets clearer than that. So it's very important to understand that all the industries are connected, and I particularly like that quote where you said that we all swimming in the same gene pool, and this is absolutely true. And also very important to understand the fact that like antimicrobial resistance is not one thing, but there is like thousands of different ways that this can apply. So it's really important to have a specific solution to tackle a specific problem. Thanks for that, Neil. And let me dig into the question. So I want to start with Maya. Okay, so we start a little bit slower, and we start with a little bit of a general question that was actually asked by one of the customers. So what is happening now in the scientific community in terms of antimicrobial resistant research? So what are the hottest topics that you're working on?
Yeah, thanks for the question, because right now I think it's a very exciting time in the sense of, let's take, for example, this quote, the quote that you just mentioned, that we all swim in the same gene pool. And in this quote, the interesting thing is this gene pool, which means that there are genetic fragments or genes that can be transmitted between bacteria, animals, humans, and there are now unprecedented methods to actually identify those genes and to really classify them, to track them, and to quantify them. And so our research has been focusing a lot in these new technologies to use sequencing, so like identifying the sequences of these genes in a very fast and very accurate way, to be able to also track how these resistant genes are not only transmitted, but also in which quantities do we find them. So in Biomin, we have been developing also technologies where we can take sequencing devices on site to perform analysis really directly at the farm and investigate which genes are present there, in which amounts, and also link this to which bacteria are the main carriers of these genes. So I think in this direction, there is a lot of development in the sense that we are switching a little bit from culture-based methods that have been traditionally the way to identify resistance, and which can sometimes help us a lot, but they are relatively slow. They are actually, they can become quite expensive. Shifting from that to more of molecular diagnostics, which can help us be much faster and also more accurate. And so in this line, we are doing actually quite exciting research. On the other hand, we also are looking at monitoring and prediction. So can we look at what are patterns around the world and try to predict in which regions we get increased or decreased not only antibiotic resistance, but also in the field of mycotoxins. So can we build up databases and prediction models that help us understand when are raising or lowering levels of mycotoxin risks in specific areas. And I think there's also quite some research, especially because we know about threats of emerging mycotoxins. So in that field, we are also performing research, especially looking at big data, which is also now an additional component of our research, not only in the lab, but also analyzing big amounts of data.
Thank you, Maya. Wow, lots of things cooking. I remember about this it's a gene sequencer, right? It's like the size of a USB stick. And you can really go to farms and like have a almost real-time response on like what is happening in the animals, what are the resistant genes, what can we do. So actually, we can tackle those challenges in a quite specific way. Myself, together with Laurent, we are in the Microtoxin risk management, so we are well aware of the mycotoxin prediction. This is something that's been deployed already a couple of years ago. It's something that is becoming bigger and bigger, and it will probably be the strategy of the future. Although it doesn't represent a way to assess whether you need a mycotoxin risk management solution or not, but still it can give you an idea of what to expect in the next year. So like very, very powerful technology, very good, very interesting way of using data.
You're muted, Michele.
Oops. Could you hear everything I've said, or was it just now?
Just the last part.
Oh, I don't know how that happened. Sorry about that. The beauty of being live. So I was going to say, I have a question that is like a little bit general for the whole panel over here, you know. But maybe I start with you, Maya, because you were already making a little bit of an introduction of the new topics. So, what are the main factors that are affecting animals when we are reducing or replacing antibiotics in feed? I mean, replacing is more something that happens in Europe. In Asia, we are talking about antimicrobial reduction. So if you want to start, and maybe Neil or Laurent, you want to build up on that. Thanks.
Yeah, well, we with the research that we are doing, we have looked at solutions for these. We cannot really say replacement completely of antibiotics, but at least reduce as much as possible the use and combine different methods to obtain the same results as when we remove the use of antibiotics. So our research has shown that when we reduce or remove antibiotics and supplement this or combine this with feed additives or also, for example, acidifiers, that actually we can maintain very similar or even improve levels of performance, gut health, and this doesn't necessarily has an impact on, for example, lowering increasing the feed conversion rate, for example, or lowering performance. So we know that there are alternatives, and these alternatives have to be additionally then combined with a good management in the farm, combined also with biosecurity, vaccines, and so that we know that there are solutions where we can reduce or replace the antibiotics with these other new and known products.
Yeah, fully agree, Maya. And in terms of, I think it's also management now becomes much more crucial in terms of this. So it's understanding all of those things in the chain, the biosecurity, the environment, And there are many farms around the world that are completely antibiotic free. And depending on the consumer's requirement, that may well be the driver. But as I mentioned, from a welfare point of view, we should not be focusing on being completely antibiotic free. It's antibiotic growth promoting free, I think, is the important thing, because antibiotics, like in human medicine, are really important. And from a welfare perspective, if we have a sick animal, we have to have a strategy of making that animal get better. So we don't want to be antibiotic free. We just necessarily, well, not necessarily, we want to be reducing the use and the reliance. And I think over the years, people have realized that the antibiotics have been a little bit of a jab or a bit of a feed and you solve all your problems. And I think as we've got modern animal production methods, people are realizing now that antibiotics have been not as effective because they've been overused. And if you actually improve on your management and your biosecurity and your hygiene, all these sorts of things, and reduce the requirement for using antibiotics, you can actually get better performance. And certainly from my European colleagues, they would have seen over many trials that have been done now that the AGPs are having less and less effect. They're not as active now, and many people have actually got rid of AGPs simply because they weren't working anyway. So I think this is, Michael, there's a bit of an evolution in terms of understanding what are the drivers within our animal production systems and how we actually need to implement various strategies at various times of the animal's life to optimize their performance and their welfare.
Understood. Thanks a lot for your comment. And yes, indeed, it's very reassuring to see that we can gradually, move away from antibiotics and growth promoters, and with the research that there is behind feed additives now in general, I mean, we can see actually the same performance slowly building up and really reaching the level of that of using growth promoters without having all the extra problems. I have a question for Laurent now, as a mycotoxin risk management expert. So, if you're talking of an antibiotic-free production system, are the effects of mycotoxins more evident?
Well, regarding mycotoxins, what can we expect? Mycotoxins are the main target organs are liver and the intestine, which are the most important organs for the immunity in the body. So this is already an indicative for us that mycotoxins they play a major role when you talk about immunity. So when we have an antibiotic-free production or a reduction of antibiotic, we have to have many different strategies to try to mitigate the risk of diseases. And what happens with mycotoxins, because we're not talking exactly about the virus or the bacteria, but mycotoxins, they can have this toxic effect that can induce to have more diseases in the field. So it's important when you have antibiotic-free or a reduction of antibiotics in your production that you always think as mycotoxins as also a predisposing factors to other diseases. And as we want to prevent it in the field, we also want to have this protection against mycotoxins. I would say that when we have this reduction of antibiotics, yes, the effects can be more evident because the animals, they can be more susceptible to any kind of diseases. So we have to take care when we are having this reduction of antibiotics also to have a prevention of the mycotoxins in the field.
Indeed, thanks, Laurent. So it's very important to stress that mycotoxins, they do predispose the animals to have more diseases, right? So even especially if you're going into antimicrobial reduction or even like a complete replacement. And I want to stick with you, Laurent, for a minute, because I actually got an interesting question from one of our customers. I got asked, so so can the mycotoxins help to mitigate the effect of bacteria and viruses? So can they consider antimicrobials? I think the reasoning behind our customers is that if these mycotoxins are poisonous and they can kill bacteria, maybe they can help and they can kill also the bad bacteria. It is a legitimate question. I think we all know the answer to that, and there is plenty of research that shows that, but it is a legitimate question. So I would actually like to hear your take on that.
Yeah. When we say, is this an antimicrobial or not, we cannot consider exactly as a direct antimicrobial, but because mycotoxins, they are not considered as microbials. They are secondary metabolites of the fungi. So when we think about the prevention of mycotoxins, this is an indirect effect, because once you have mycotoxins, you are going to have the reduced immunity. I can give some examples here that are very interesting when you talk about bacteria or virus. For example, we have DON, that is deoxynivalenol, that is one of the main mycotoxins that we find in the feeds all over the world. DON has an effect, the mode of action that in the intestine, it goes into the velocities and destroy the velocities. And this makes that the bacteria, they can be more present in the blood circulation. This makes that the animals, they can develop more bacterial diseases. This is one of the examples. And for example, another example regarding also DON is that there are many studies that they say that when you have DON present, you also have this reduction of the vaccinal efficiency. So if we have a virus, for example, and you do not have the complete protection of the vaccine, you're going to have problems that are not exactly directed to mycotoxins, but you have this indirect effect. So whenever we think about antimicrobial, in a combined effect, we would have the need of antimycotoxin product to be together.
Yes, I totally agree, Laurent. And I think this is what, as I mentioned, I think the customer intended, okay, you know, to mycotoxins work a little bit like an antibiotic so that they can kill the pathogens, but we have plenty of evidence that this doesn't happen. If you think about all the studies, like you said, with DON, necrotic enteritis, coccidiosis, salmonella, and pigs, there is plenty of evidence that actually mycotoxins, they work together with those pathogens and make them more severe. Thanks for your take on that. I have a question for Maya and Neil. Those are actually two separate questions, but maybe we can group them together in one. So, what are the current solutions applied for fighting resistant strains? So maybe Maya, you can tell us a little bit from the research perspective, and maybe Neil, you can give us a little bit of a field overview.
Yeah, well, we know that there are several technical feed additives that can be used to combat these resistant strains. And of course, that the first thing to reduce the amount of resistant strains is to use less antibiotics. I mean, we know that there is a clear relation between usage and the amount of these strains. So the first thing, of course, reducing the usage so that now we can combat and reduce those resistant strains. And on the other hand, we can use these feed additives that not only have an effect, a positive effect on the health of the animals, but also we have seen that several of these feed animals can target specific bacteria, pathogenic bacteria, and reduce the levels of those bacteria. For example, E. coli or Campylobacter. So we know that using these feed additives, probiotics, we can reduce the populations of these bacteria. And the interesting thing is that many of these pathogenic bacteria are the ones that carry most of these genes and carry pieces of DNA that can be transmitted to other bacteria. So reducing those specific pathogenic bacteria that are the hubs of where these genes are concentrated, then we can definitely lower then the not only resistant strains, but also lower in general the gene pool, the resistant gene pool.
Yeah, absolutely. And in terms of the field situation, we want to make sure that we're killing the bacteria with a feed additive, but killing all of the bacteria. And there's been lots of evidence showing that not only would we reduce, say, E. coli counts when we use feed additives, we also reduce the amount of resistance genes that are left in there. You don't want to apply a feed additive that kills the majority of bacteria, but allows some resistant bacteria to survive. And this is at the essence of the zinc oxide ban that's will shortly be in place in Europe, is that inadvertently or directly, zinc oxide use at high levels, at the 3 kilos per ton, will cause some selection of bacteria to grow, and those bacteria that survive also carry a lot of resistance genes. So this is why, one of the reasons, in addition to the environmental issues of zinc oxide. So when we're using feed additives, yes, we can have a direct killing effect on the bacteria, but we also want to promote good gut health in terms of stimulating the lactobacilli and the other bacteria that will actually outcompete the pathogenic bacteria. And at the same time, promote the immune system of the animal to be optimized in terms of responding to a challenge, and also not consuming up too much energy in terms of having the inflammation system and the immune system always turned on. So there's a number of strategies, as Maya identified. We want to reduce those pathogenic bacteria in the first place, but we also want to support good gut health so that the animal helps itself to combat the pathogenic bacteria. And by reducing the pathogenic bacteria, you also should be reducing the amount of pathogenic bacteria containing resistance genes. So this is the general strategy. And the rest of the next half hour, we'll try and get down to some of the detail, but this is the general strategy.
Thanks, Neil. Actually, I received one of the questions about zinc oxide, but maybe I will ask you a little bit later. But yeah, very, very important take-home message is focusing also on reducing the gene pool so that we can shut down those resistant genes once and for good, and maintaining gut health. This is also crucial. And I want to take this gut health concept and move a little bit to Laurent, because we know that gut health is also heavily influenced by mycotoxins, like at the beginning you asked and you said. And there is actually a question that goes with how important is Microtoxin Synergies when it comes to animal health and gut health in general.
This is a very frequent question that we have in the mycotoxin field regarding the synergistic effects that we have in mycotoxins. Because when we have a feed, normally more than 60% is contaminated not just by one mycotoxin, but less than one. Normally we have a multi-contamination about mycotoxins. And there are two different effects that we can have, the addictive effect and the synergistic effect. What does it mean? In the additive effect, we have one mycotoxin plus one, we have two effects of both of them. When we have the synergistic effect, we have 1 plus 1 can be 3, 4, or 5. What does it mean? When we have more than one mycotoxins, we can have this combined effect that makes the toxic effects of the mycotoxins even more important for the animal. When we go into a normal situation in a commercial farm, what we see? This multi-contamination, we have this additive effect and also this synergistic effect happening at the same time in the field. So this happens in the animals, and what can we see and expect of this whole situations? That even in low levels of mycotoxins, that normally we would go, if we just look for one mycotoxin, we would sometimes not even consider that important. But when we have more than one mycotoxin happening at the same time, we can see that the synergistic effect can make a really big challenge in the immunity of the animals. What can we expect of these situations and how to identify mycotoxins like this? We can see that we have these recurrent diseases that are happening in the field, or we have this low effect of the vaccination, and also it means that consequently we are going to have a lower performance that we would be able to reach if we do not have a mycotoxin, or if we have a good mycotoxin risk management strategies. So this is very important to know that in the field we have these low doses, but they can work synergistically and additively. And this is important to always think, how can we counteract all of them and having great strategies for the most important mycotoxins.
Thanks, Laurent. I totally agree with you, and I think what you said is it's really important. And we have the problem in Asia, not in all markets, but we have it in quite a few markets where people they just tend to focus on one mycotoxin only when it comes to management, and this is usually it's aflatoxin because it's the most dangerous carcinogenic mycotoxin. Is also the most famous one, the most popular one. But it is actually important to focus on the cocktail and on the combined effects that those mycotoxins can have on gut health. And I think we connect a little bit with what Maya and Neil said before, right? So fighting the resistant genes, fighting the bacteria, and also clearing up the gut and the animal, the whole animal, from all the other stressors that come from outside. So thanks for that. And moving a little bit into the direction of more formulation of modern feed additives. So I have a question for you, Maya. Where do the microbials that we use in feed additives, the probiotics and everything, where do they come from?
Yeah, well, these antimicrobials are actually a main, they occur by nature in several bacteria. We know that this exists since a very, very long time. And bacteria use some of these molecules or some of the products of these genes to counteract their own, let's say, to avoid that they get killed. But the thing is that when we start to use these antimicrobials in very, very high amounts and without any specific control, then we are selecting for those bacteria that can survive, and these are then becoming a problem because those antimicrobial genes can be transferred, and they become, instead of being a natural mechanism, it is now transmittable and an acquired resistance. And this resistance can jump into pathogenic bacteria, and this is what's causing the problem. So they are normally, they were at some point normally in the bacteria, but now it has become more of a problem because of this spread and acquiring of resistance.
I think the question was a little bit in a different direction. I think it was more when you are developing a feed additives, when you have to investigate new probiotics and so on. Usually, where do they come from? They come from the animal directly. Do they come from the environment? So the probiotics that you find in feed additives.
The probiotics that we are using in the feed additive. So we are trying to identify these probiotics from the specific animals where we want to apply them. So if we want to have probiotics for the poultry, for example, we from our research can isolate then positive or beneficial bacteria that are derived from poultry. And then these ones are then in the lab investigated, purified, and then these ones are then used later on as probiotics and in feed additives. So they are also similarly for swine. We can go to bio or organic farms, try to get from the animals some samples, and for example from the milk, or for example from other parts of the animal, and we are investigating the properties of these potential beneficial bacteria, and then select from those the best to be then incorporated in the feed and used as probiotics.
Thanks, Maya. A follow-up question for you and maybe for Neil. Is there any negative effect of using those probiotics in the animal, for instance, increasing FCR or any other negative effect that you could think of?
I can start by saying that there are many trials where we have seen no negative effects. On the contrary, we see that these feed additives and probiotics are helping with gut health, and also that there is no negative effect on the feed conversion rate or on the growth rates, for example. Actually, we have seen very positive and beneficial effects in the use of these probiotics with very good, even improved performance and also growth rates.
Yeah, and from the field perspective, Michele, you know, there's no reason why a probiotic should make the animal perform any worse, but certainly we have to make sure that we apply any feed solution or feed-based solution to the right challenge. It's a bit like when you use a fire extinguisher. You know, we have fire extinguishers for wood, we have fire extinguishers for petrol. If you use the wrong fire extinguisher, you could make the product, the fire go worse. And so while there's no evidence that we have, you know, probiotic will make the situation worse, but you have to use the appropriate tool at the appropriate situation. So I think that's important, because sometimes people have in their head that I've got this issue, I use this product, but you've got to make sure that you match the product and the features and benefits of that product to the particular challenge that your animals are experiencing at that time. And importantly, as the animal grows, you know, if it's a chicken, it goes, you know, becomes a pullet, which may lay eggs as a hen, or as a sow, it produces piglets, which become grower finishes. You have to use the right solution for those different phases. And so the wrong probiotic used at the wrong time, it won't make it worse, but it just won't give you the result that you need. So again, it's about choosing the right solution for the right product at the right phase of life of the animal.
Thanks, Neil, and also thanks, Maya, for your answer. Yeah, I fully agree with you. And sticking to what you just said about using the right tool for the right job, Laurent, I want to ask you a question about mycotoxins. And this was asked by one of the customers. So, what would be your advice on how to incorporate a mycotoxin strategy into an antimicrobial reduction plan?
This is very interesting, because when we think about mycotoxins, we are always thinking about a predisposing factor to diseases. So when we have mycotoxins, this overall mycotoxins, I'm not saying just about one, as we talked before, we have to think about all of them. So it's very important to have a prevention of mycotoxins as they are normally found in the feed. So having a mycotoxin risk management strategy very well defined. When I talk about mycotoxin risk management, I'm talking about identifying which are the main mycotoxins that we have in the feed, that we have in the region, and in the compounds that are used as ingredients for this feed. Having this an analysis that is going to say for us which are the mycotoxins that are there, and having the right counteracting strategies for each species and phases. This is important to really look forward into what are the challenges and which ones we have to focus on. So when we have these other antimicrobial reduction plan, we also combine it with a prevention of mycotoxins. What I would say is that we have to be very focused in which would be the phases, the animal and the species that we are looking forward to have this strategy established.
Yes, no, and I totally agree with you, and I think you are clearly going in the direction that where Neil and Maya were going. So using the right tool for the right job, I think, is essential. So when we are using feed additives, this has to be strategized. Animal and feed producers, they have to request for the help of our experts, because we can guide them in the solution process, and we can in the selection process, and we can guide them into getting the best out of the product that they're purchasing. So we are running a little bit over time, so I will probably not be able to cover all the questions, but don't worry, ladies and gentlemen, because whatever we cannot answer here, we will send you an answer via email. So, but for the last part of this webinar, I want to ask a few open questions to the panel. So let me start with the first question, and it's: if humans do not eat meat, are they still susceptible to resistant organisms? Maybe Maya, you can start if you want, and later Neil and Laurent can build up if they wish.
Yeah, so this is a question that we get actually quite often. Why do we have to focus on managing or reducing antibiotic resistance in farm animals if I am a vegetarian, for example? And as Neil already showed this very nice schematic at the beginning, we need to focus on antibiotic resistance reduction as a fully linked process. It's that's why we call it this One Health approach, where you know that even if you don't consume dairy or animal products, the flow of genes of resistance is happening every day overall. So genes that are present in animals will also be able to make their way through to crops and to the water, and we drink the water. We also get sick, visit the hospitals, we acquire there also some other resistant genes. And so it is really this holistic view where everything is interconnected, and we cannot really separate or say, if I don't consume this, then I am protected. So we have to realize that basically everybody is susceptible, and we have to look at it in this more interconnected approach.
Neil, you're muted.
Absolutely, and I guess just to add, you know, it's the appropriate and prudent use of antimicrobials, both in companion animals and humans. So even if you're a vegetarian and don't eat meat, you may well be prescribed a course of antibiotics, and you have to, you know, use that antibiotic course appropriately, because if you just use it for a day or so and you feel like you're feeling better, you've maybe just knocked on the head of some of the bacteria, but you haven't actually eliminated those ones. And so, as Maya indicated, we know from our studies and many other literature studies, when you use an antibiotic, you massively increase the antibiotic resistance in the remaining population. And so in our cats and our dogs, we have to make sure we use the appropriate antibiotics to make sure we eliminate the bacteria that carry those resistances. And to some extent, even though you're not in contact with a farm animal or eating the meat, the companion animals are actually probably a much more significant source of antimicrobial resistance, because they're in your house, they're on your food surfaces, you're, you know, close proximity to them. So the bacteria that are in the cat and the dog are very rapidly transferred to the human. And any resistances that those animals pick up will be transferred to the human, whether they're a meat eater or not. So again, it's all about appropriate and prudent use of antimicrobials.
When I say all the comments, I agree a lot with you, Maya and Neil, regarding that this is One Health approach. And we have to look, when we think about reducing the antimicrobials, we have to think about all the other factors that are connected, and how to reduce also the predisposing factors. And they are all of them connected. And what is the idea here? Is to increase the immunity for the animals and also for the humans to be more able to fight against the other problems with bacteria and virus and also mycotoxins that they are. This is going to be a hazard for the organism. So thinking about One Health, we have always to look this in this holistic field with all the factors, the main factors, but also these predisposing factors to have this antimicrobial reduction.
Yes, yes, indeed. Very good comments from all of you guys. There are a couple more questions that I want to ask, and I have 5 minutes for that before the closing questions. Okay, so I want to remain a little bit on the last question and have a follow-up. So the follow-up would be, how fast does antimicrobial resistance spread? So we are talking about farm level, and we are talking about a very practical example. So if we are detecting antimicrobial resistant genes in a farm when we are sampling or something, is that representative for the whole region? or not? So what is the significance of those data? Should everyone panic, or this is something that is likely to remain in one farm? This is actually an open question, but maybe Maya, if you want to start, and then the other two speakers can also build on that.
Yeah, well, we have been monitoring some of these antibiotic-resistant genes in different countries, specifically focusing at the moment in Europe, but we want to expand this. And we usually see that that there is quite some variation between one farm and another farm of, for example, the same country or the same region. And I think this is at the moment still relatively linked to or directly linked to the usage of antibiotics in specific farms. But we also need to see that different countries have different regulations and different usages of antibiotics, and this is also then linked to the amount of resistance that you are able to see in a farm. So on one hand, we see, yes, there is variation between farm and farm, and this can mean that that only by looking at specific farms we have a better understanding of how these antibiotics resistant genes spread. But on the other hand, we know that there are patterns where a specific country, because of the legislation, has overall a higher amount of resistant genes than other countries in the nearby region. So yeah, there is variation, but there is also certain patterns that we start to look at, for example, country or region levels.
Yeah, and just to add from the farm level, Michele, it's important to look at the techniques that we use in Maya's lab. That they're looking at the next generation sequencing. Very high level thing that you look at the resistomes, the actual resistance genes within the bacterial population. This is at the moment not available for every farm. You just can't walk onto a farm yourself and do it. So what the farmer is using is often back to our old techniques of culture. So the veterinarian will go in and there's a disease, and ideally the veterinarian will take some samples and do the good old minimal inhibitory concentration test. So look at what antibiotics should they use to address that. And so it's a combination of the modern technology, what we see in Maya's lab, which maybe in 5 years, 10 years will be commonplace, and everybody can just take a sample, run it down to their local laboratory and have a complete resistome done very quickly. So at the moment, it's very difficult to look at what are the resistance patterns we see at the lab that Maya's doing and other centers around the world, and what the farmer is seeing on their own farm within. But as Maya said, and we see this, when people are measuring it, and I think that the important thing is you've got to be able to measure something before you can manage it. But we do see when you use particular class of antibiotics, resistance generating in those particular areas. And this is when you start getting into the detail around what antibiotic classes have been banned, what have been phased out, and what are human relevant or various other classifications. Coccidiostats are an antibiotic in theory or chemically, but have low impact in terms of human relevance. So again, there are different antibiotic classes, but it's more important that we focus immediately on the ones that have human relevance in terms of developing resistance, and at the same time reduce our reliance on all antibiotics wherever we can, using management, feed additives, hygiene, etc.
Thanks, Neil. Yes, I totally agree with you, and I think this is a little bit what the question, the direction of the question. So people want to understand if there is a problem for them or a potential problem for them. And this actually brings me to the last question, because yes, we are talking about this resistant microbes being able to transfer to humans, but what about mycotoxins, Laurent? So can mycotoxins be carried over to humans at dangerous level? What do you think? From animal products, of course.
Yeah, most of the mycotoxins, they have these metabolization and excretion that they're very fast. So most of the mycotoxins, most of them, they do not have problems with being transferred to the tissues. this is not such a major problem for the main ones that we know. The major exception that we have here is for aflatoxin, for example, because for aflatoxin, it can be transferred to the milk. In the rumen, it can be transformed by aflatoxin B1 into M1, that this is a very carcinogenic aflatoxin. And this is very important when we think about the population, the part of the population that most consumes milk, that are the children. So this is very well regulated normally by the countries, and having all these legislations that we have very looking forward short thresholds for the mycotoxin, for aflatoxin, for example, in milk. But for most of the mycotoxins, this is not such a big problem to be transferred into the byproducts of animals.
Thanks, Roland. And yes, I totally agree with you. And aflatoxin being the major problem because of the carryover into the milk. So we have 3 minutes to go, and I would like to ask a closing question, and this is really open to all the panelists. How do you see the trend for antimicrobial resistance in the future, especially now, we are having an increasing population, increasing demand of protein. So do you think antimicrobial resistance will become even a greater problem in the future, or do you think the industry, our industry, especially feed additives and everything, they are doing already a good job to contain this? I know in Europe already antibiotics are banned in several countries, but for instance in Asia we are not quite there. So what is your general take?
I might make a start first then.
Yes.
Yeah, I'll make a start. Look, I think as I mentioned that, it is an important consideration for everybody. And certainly, as our tools enable us to measure AMR more importantly, and as various other countries start to look at, not only the kilograms of antibiotic used, but the kilograms per meat animal produced or milk produced or eggs produced. These are some important metrics that the governments are trying to get their hand around. And so measurement is really important, but because we only have a fixed number of antimicrobials available to us as humans and for animal welfare, it's not a trend for antimicrobial resistance. It is a real problem, and we need to continue to work hard at it, because bacteria replicate. By the time we started this seminar to now, the bacterial populations have probably grown 100-fold. So you can see that over millennia of bacteria evolving, this resistance, the bacteria will outsmart us. We're seeing COVID warp all the time and coming up new variants. So the bacteria will continue to develop resistance to every known chemical that we can throw at it. So we have to look at our approach and change our approach, be much more holistic around how we want to tackle it. But it won't go away, but we can, firmly believe we can manage it and keep it under control once we have the right techniques through the labs like Myers, and once we have the right application on farm, considering management, environment, hygiene, as well as feed additives and vaccines. I think the trend, if you want to call it a trend, will become more significant, but we do have the tools to manage it. And awareness, I think, is the key thing. We've got to make sure that all of our animal producers and all the supply chain are aware of the issues and how do we manage it.
Thanks, Neil. Maya, Laurent, you want to add something?
I think there's not so much to add to this, what Neil already said. We just need to think positively with the data we have. We know that the process is reversible. So the increase in antibiotic resistance can be reduced by all these approaches that we have been discussing about. So we have to also see it in a positive way, in the sense that we know it's increasing, but this is completely reversible. So we have to work towards that.
Absolutely.
Just to add to both, I think that it's very nice that we already have this awareness and we have this conscientious that we cannot go forward using the antimicrobials as we were using before. And I see, as Maya and Neil, that this is an integrative process, and we have technology to look forward it and make development of new strategies to reduce this resistance.
Yes, I totally agree, and I think it's important to close this on a positive note. And yes, we are doing a lot of research. We are working on that. The process is reversible. The industry is doing a lot of work into coming up with new solutions to tackle this issue, and also to raise the awareness and to work on that on a holistic concept. And already now we have plenty of solutions to target gut health, to target antimicrobial resistance, to target mycotoxins. So yes, definitely the future is positive. At least I believe so, and I want to believe that. So that brings us to an hour, and that brings us to the end of this webinar. So I hope that your questions were answered. We really appreciate your time in submitting those questions, and hopefully we will have, if the feedback is good, we will have more events like that in the future. I think it's important to break down this concept and to make it easier for everyone. Everyone should have a good understanding on that. Without further ado, I would like to thank the panelists for joining this beautiful discussion that we have. And I wish everyone a nice evening and a very nice weekend. Thanks for your attention.
Thanks, Michele. Thank you, everybody, for listening.
Thank you. Bye-bye.
Bye.